Good morning, dear students. Yesterday, we learned regarding one of the cell organelle which is only present in plant cell. So, those are plastids. So, in plastids, we have three types, that is chromoplast, leucoplast and chloroplast. So chromoplast is for the coloring the other parts of the plants other than green. Leucoplast usually it will be related to the storing organ or a storing plastids. For example, radish. Radish is white in color. So leucoplast will be present in the radish. Next one is chloroplast. Chloroplast is a plastid which forms a pigment called as chlorophyll, which is the important component for the photosynthesis. So yesterday we studied the structure of the chloroplast. Here you can see the outer membrane, granum as well as lumen, and lumen means the place, and as well as the stroma, that is a fluid, and a coin like structure called thylakoid, and outer and inner membrane. So this is the structure of the chloroplast. In today's class we are going to study regarding endoplasmic reticulum and other cell organelle. So online classes of today's had been done already. The recorded was being done, but due to technical issue audio will not was not able to record it. So that's why I am doing one more time for the YouTube viewers, YouTube students. So here we are going to study the next cell organelle, that is endoplasmic reticulum. So why it is called as endoplasmic reticulum? So as we all studied regarding the cytoplasm. So regarding cytoplasm, when we studied, cytoplasm is a fluid. Here orange colors, light orange color, whatever you are seeing the fluid, so that is the cytoplasm. So the cytoplasm, the outer layer that is near the plasma membrane, it will be thin and transparent. When it will come near the nucleus, it will be quite thick. So the outer cytoplasm is called as, that is just before the plasma membrane, it is called ectoplasm, which is near the nucleus or around the nucleus, it is called endoplasm. So the structures which forms the network in the endoplasm are termed as endoplasmic reticulum. So when you see the image, you can see these are a network like structure. So these network like structures, they will be attached to a nuclear membrane or the plasma membrane. So you can see that the sac-like structures, so these sac-like structures are called as cisternae. Okay, this cisternae, sac-like structures, and these, when you see the light orange color, so these are like tubes. These are called tubules. And in some organisms you can find a spherical structure of endoplasmic reticulum which are called vesicles. So we have three components or three kinds of endoplasmic reticulum: cisternae, which are sac-like, tubules, which are tube-like, and a spherical, which are vesicles. Here you can see a certain pink color, right? So on the pink color you can see a small spots, a spherical structure, which are referred as ribosomes. So the endoplasmic reticulum, which has ribosomes on its surface, is termed as rough endoplasmic reticulum. Why it is termed as rough endoplasmic reticulum? Because rough endoplasmic reticulum, what it makes the surface rough. Understood? Now when you see this mild orange color, so here the mild orange color, it does not have any ribosomes on its surface, so its surface is smooth. So this kind of endoplasmic reticulum is termed as smooth endoplasmic reticulum. So we have two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. The next image whatever you are going to see, it is a image of endoplasmic reticulum in a electron microscope. It is a micrograph, the printed image which we got from the electron microscope. So you can see rough endoplasmic reticulum where ribosomes are attached, as well as smooth endoplasmic reticulum. Next, what are the functions of endoplasmic reticulum? As these endoplasmic reticulum, these are as a channels, these act as a channels, so they are involved in transportation of materials from the nucleus to the cytoplasm or from the cytoplasm to the nucleus. And as they have ribosomes, they are involved in the secretion of the proteins. Ribosomes are called protein factories. So as rough endoplasmic reticulum has proteins on it, they are involved in secretion of proteins. Then smooth endoplasmic reticulum. Smooth endoplasmic reticulum helps in the detoxification. So what do you mean by detoxification? De means remove, toxification means poison or toxins. So it helps in a detoxification of drugs or any toxins which has directly or indirectly entered our cell or body. Next, smooth endoplasmic reticulum. You know that a plasma membrane, we learnt a fluid mosaic model of plasma membrane. Here the plasma membrane is made up of lipids. Okay, one of the component is lipid here. So these lipids are synthesized by the smooth endoplasmic reticulum. So what these lipids will do? These lipids helps in the formation of the plasma membrane. So these lipids are formed by the smooth endoplasmic reticulum. So we can say that smooth endoplasmic reticulum helps in membrane biogenesis. What is membrane biogenesis? The formation of the membrane using the lipids formed by smooth endoplasmic reticulum. Next, and also the endoplasmic reticulum also helps in the transportation of hormones or any other substances within the cell, not outside the cell. Within the cell, if any substances has to be transported, it will be done by the endoplasmic reticulum. Next, the next cell organelle is Golgi apparatus or Golgi complex. Why it is termed as Golgi? Because it was discovered by one of the scientists named Camillo Golgi. So here we have again the components of Golgi, that is in Golgi complex some of the structures are sac like what you are seeing. Okay, some of the structures are spherical and some of the structures will be tube like, or some of the structures are very circular. So we have cisternae, tubules, vesicles and vacuoles in the Golgi bodies. So these Golgi apparatus will be floating in the cytoplasm. So when you see the cis phase and the trans phase, so the cis phase is the phase which is facing towards the plasma membrane. Trans phase is the phase which will be facing towards the nuclear membrane. So here you can see here. So the cis phase, this is a cis phase which is facing towards the plasma membrane. That side is a trans phase. So whatever hormones has to be secreted or has to be transported, those vesicles will be formed near the cis phase, that is a formation phase, and it will be transported to the trans phase, that is maturation phase, and from the trans phase the vesicle will be goes to the targeted part or a targeted organelle with a respective secretion. So Golgi apparatus will be involved in storing, packaging as well as transporting the materials needed. For example, hormones and proteins. So these all will be transported by the Golgi apparatus. Hence Golgi apparatus is termed as traffic police of the cell. And Golgi apparatus is also involved in formation of lysosomes. What are lysosomes? Lysosomes are also the cell organelle. So we will learn its function in the other concept. Next, the Golgi apparatus, as I said, it helps in storing, packaging and the moderating the chemical substances or the hormones, and it also plays a crucial role in the formation of the glycolipids and glycoproteins. So what do you mean by glycolipids and glycoproteins? So you saw those terms in the plasma membrane. So the substances which are needed to form the membrane, that is glycoprotein. Glyco means carbohydrate, protein means protein. When carbohydrate attached to the protein combines with the protein, how they will combine? With the help of the Golgi complex. So Golgi complex helps in the combination of the carbohydrate and proteins as well as carbohydrate and the lipids. Okay, so in plant cell, what happens? In plant cell, the Golgi apparatus will not be like this. The components will not be attached like this. Those will be scattered. So there in plant cell, the Golgi apparatus is termed as dictyosomes. Those are not termed as Golgi apparatus. In plant cell, we will refer it as dictyosomes. Okay, so this is the micrograph of Golgi apparatus, where you can see the cisternae, and these are the tubules as well as vesicles. So this is regarding the Golgi apparatus. Next, so we learned regarding the ribosomes. So ribosome structure is spherical in nature. These ribosomes will be attached to the endoplasmic reticulum, or also you can see these ribosomes will be floating in the cytoplasm also. So what these ribosomes will do? These ribosomes are involved in formation or synthesis of proteins. So as they synthesize proteins, ribosomes are termed as protein factories of a cell. So this is regarding the ribosome. Next, one more cell organelle called as mitochondria. So when you see the structure of mitochondria, it is made up of two layers, that is outer membrane and inner membrane. So in between outer and inner, we will be having the space called as intermembrane space. So inside the mitochondria, the fluid will be present, which is termed as matrix. The inner membrane will be folded. You can see the green color structures. So the inner membrane will be folded and it is irregular, forming a structures called as cristae. So on this cristae you can find a small particles or small structures, dot structures. So these structures are called as F1 particles or Racker's particles. So what these Racker's particles or F1 particles will do? These are involved in synthesis of energy currencies called ATP. So what do you mean by ATP? Adenosine triphosphate. adenosine is a nitrogenous phase and tri means 3, phosphate means phosphor. So adenosine triphosphate are the energy currencies where the energy will be stored. Whenever we need the energy, the ATP will be broken down and the energy will be released. That is why the F1 particles of mitochondria are involved in the synthesis of ATP. That is why the mitochondria are called as powerhouses of the cell. So even the mitochondria has the ability to replicate, to produce itself. One mitochondria has the ability to produce the other mitochondria. How? Because it has a DNA, its own DNA. So it has a capacity to produce the its own copy. So this mitochondria is referred as semi-autonomous organelle. We have one more semi-autonomous organelle, that is chloroplast. Chloroplast will also be having its own DNA where it has a capacity to produce its own copy. So these two cell organelle, that is chloroplast as well as mitochondria, are referred as semi-autonomous organelle because they have a capacity to produce their own copy due to their own DNA. So here we completed the cell organelle like the plastids, endoplasmic reticulum, mitochondria, ribosomes and Golgi apparatus. So in the next class we will be continuing with few more cell organelle. So online session recorded was not able to upload it on YouTube because of the audio problem. So I recorded one more video so that the YouTube students will get through it. So I hope the class was good. I hope you understood. If you got any doubts, please ask me on an online session. Thank you one and all. Have a nice day.